I'm Wendy Mendel-Rubenstein, a board member for Exposomics and Children's Environmental Health at Mount Sinai. Thank you for joining us for this week's lunchtime chat in our Environment and Health series. We hope today's session will provide insights into the groundbreaking and inspiring environmental health and exposomic research happening at Mount Sinai. When we talk about exposomics, we're referring to the totality of an individual's environmental exposures, starting from conception and throughout life. Many exposomic and environmental health scientists are studying the exposures due to our changing climate and developing interventions to protect the most vulnerable from disease. Among those scientists, and one of the world's most prominent, is Dr. Rosalind Wright, and it is my privilege to introduce her today, especially on International Women's Day. It is wonderful to be able to introduce a leading female scientist. She is going to discuss how her important work in exposomics will lead to breakthroughs in preventing lung disease, especially among those who are most vulnerable. Dr. Wright is an internationally recognized clinician scientist and epidemiologist. She is co-director of the Mount Sinai Institute for Exposomic Research, dean for Translational Biomedical Sciences, and the Horace W. Goldsmith Professor in the Departments of Pediatrics and Environmental Medicine and Public Health. Dr. Wright is a pulmonologist who has devoted her career to understanding the impact of the environment on respiratory health. Her research focuses on uncovering the causes of chronic childhood diseases like asthma through the exploration of the complex interplay between social determinants of health, such as exposure to stress, poverty, and violence, and environmental toxins like air pollutants. Her findings demonstrate the importance of taking an exposomic approach that considers exposures across the lifespan and have important implications for the creation of programs and policies that improve health outcomes for all. The presentation will be about 25 minutes long and followed by 20 minutes of question and answers from the audience. Please submit your questions using the Q&A function, and we'll try to answer as many as possible. And with that, Dr. Wright, thank you so much for joining us. And I'm now turning it over to you.
Thank you so much, Wendy. You can hear me okay?
Yes.
Okay, great. It's a pleasure to be here, and particularly on International Women's Day. So I thank you very much for that shout out. It's a pleasure to be able to present some of the work that I've been doing related to chronic respiratory disease. And as Wendy said, really interested in broadly understanding the total environmental impact on programming, risk for onset of disease, and we'll touch a little bit on beyond that, how we think about it clinically to help identify those most vulnerable and decide on best treatments, et cetera. So I'm going to share my screen. Can everybody see the full presentation?
Yes, put it in.
It's on the screen. Okay.
Yes.
So I'm gonna focus particularly around.
Dr. Wright, can you move to a full screen mode, please?
It is on mine. Let's see. I'll try it again. It was showing full screen on mine. Is that better now?
Yes.
Yes.
Okay, great. Good. So I'm going to talk to you today about one component of the environment, to not to say that we only are interested in one aspect, air pollution, in relationship to how climate might change the impact of that on chronic lung disease, but just to exemplify some concepts that we want to all keep in mind when we're thinking about how complex the research is to be able to measure as much of the environment as possible, and then to translate that to how it might impact health. So really, it's to exemplify the key concepts there. And then how climate is only going to magnify some of the influences of that, and particularly air pollution. And to try to think together about how exposomics will really change the way we think about medicine and the practice of medicine and incorporation of environmental influences on our health and well-being over the life course. So as Wendy mentioned, I'm a pulmonary physician. I'm a lung doctor. I take care of adults, actually. I'm not a pediatrician. So my original work was sort of at the right-hand end of this screen, taking care of folks who already had advanced and long-standing lung disease and treating those illnesses. But I started to think, how far back do I need to go to prevent? And that takes us all the way back to the left-hand side of this panel. It really starts in pregnancy, even preconception, but let's think about during fetal development when all of our organ systems, including the respiratory system, is developing. That's one of the most vulnerable periods for environmental factors influencing disease onset. But what happens to you in the womb is really something we need to keep in mind when I'm taking care of somebody in their 40s, 50s, or 60s. And then we start thinking about, well, what are all the influences? What is the data? What does the science tell us are the factors that contribute to lung growth and development, keeping people on an optimal, healthy pathway versus when do people fall off that track? And it's all the things from your nutrition, chemical exposures. We talk about what those many things are. There are many chemicals to think about, toxic metals, others, air pollutants, psychological stress. Wendy alluded to some of the ways we conceptualize that, and where you live and what other influences you might have. And when you start to think about climate, where you live is very important because there's different impacts on various regions, flooding, drought, heat, all kinds of things beyond the air pollutant and other things. So all of these things are at the roots of the health outcomes that we're looking at. And it's some optimal balance of all of those things that can help keep us healthy. But when you start to look at the number of factors in the middle of this slide, that list even grows further when we start to unpack, well, what does the science tell us? Well, how many of these things are really influencing development of respiratory disease? And you can see the outdoor environment. There's so many components of that we need to think about, ambient air pollution being one of them. Indoor environment, exposure to things like tobacco smoke, allergens, chemicals, pollutants, molds. Again, if you think about climate and flooding, it's going to impact those kinds of things as well. Our nutrition, a healthy diet that's high in antioxidants, that's high in anti-inflammatory polyunsaturated fatty acids, things we get from fish intake, for example. We hear a lot about how that might be healthy, and that's because of these types of anti-inflammatory polyunsaturated fatty acids, or PUFA. And then psychological stress, multiple ways that we experience that in our daily lives. These are just a growing list that we like to think about. So we try to measure all of these things together in the work that we're doing at the Exposomic Institute. But let's look at the left-hand side, climate. What's going to happen, and what do we already know is happening to some of these, the distribution of these exposures, the toxicity that's related to these different factors? How is it impacted by climate change? And I was invited to submit a commentary to Lancet Child and Adolescent Health a couple of years ago to really put that together and think about influences of climate change on childhood asthma and allergy risk, because that's a particular area that I like to study. Well, we know, and we'll talk about it, air pollution. Air pollution, the distribution of air pollutant components is going to change based on climate factors like temperature, humidity, relative humidity in a region, area, flooding, etc. Allergens because of climate change. Because we have longer seasons that might be warmer and colder at different times of the year than as the time goes on, we're going to have longer periods where we're exposed to what we already know are allergenic, ragweed, pollens, these kinds of things. But it also is changing the makeup of some of the food sources that lead to increased allergy, like peanuts. So the allergenicity of peanuts has changed because of the climate effects, the different things that climate brings to different regions of the world. And that may be one reason we're seeing, for example, increased food allergies. There's an epidemic of food allergies. That's happened in a relatively short period of time. Nutrition is important. As I said, we like to have these polyunsaturated fatty acids, for example. Those protect our lungs and keep us from developing things like asthma at different points in time, and it can actually improve your outcomes for asthma, even if you have the disease at some point, by having a better balanced diet with this particular nutrient. Well, if you look at how the oceans are being affected by climate change, the balance between good and bad polyunsaturated fatty acids is changing. These are concerning things because that suggests we're going to really have to pay even more attention to what we're eating, where we're getting our food, and those kinds of things. And stress, there's lots of ways that economic strain that results from climate change, et cetera, and extreme weather events that lead to loss of economic well-being can contribute. So there, you start to see the big picture here. But let's just focus on air pollution, just one piece of it today. Let's talk about that and how we need to we have to not measure one thing at a time. We need to measure as much of it and define as much of what pollutant components that we're exposed to really understand the full scope of the impact on things like respiratory disease. That gets you more towards this idea of the exposome. Exposome, in one sense, is measuring as much of the external environment that we think is driving health as possible. Don't have to measure it all, but the things that we do know collectively contribute to our health outcomes that we're interested in. Let's do that as comprehensively as possible. So that's one way we approach it and think about it at the Exposome Center. So you think of, say, PM 2.5. That's particulate matter of a certain size, and that's largely related to traffic-related pollution. It's a big factor that we think about in urban environments like New York City, for example, other urban cities across the country and around the globe. And we usually measure it as that's the main exposure that we're looking at, and I can talk about why that is. It's because it's measurable. And we started looking at it largely because it's measurable. It's predicted by a lot of EPA monitoring that's done around the country. We'll talk about that a little bit. And we knew we could have a good measure of that. But if you look at the left-hand side of this slide, and not the pie diagram, which is the overall circle, is all of PM 2.5, but there's so many things that are making up that PM 2.5. Different gaseous components, different constituents like metals, lead, you see there listed, et cetera, but there are multiple ones there. And we've learned that we need to think about who might be most vulnerable to the different makeup of this pollution by understanding how it varies across regions, neighborhood. You can be as small down as to a neighborhood, a city, a state, et cetera, and you might look at that in different ways to say why we see more asthma in urban environments in the Northeast than we do necessarily in the Southwest, that kind of thing. So a lot of what we do, we've taken what we call geospatial approaches to looking at air pollution exposures. And in this case, we're talking about airways outcomes, but we look at a lot of different health outcomes in relationship to that. And how do we get that data? We don't go out to each individual and measure every day what they're exposed to, where they live, where they work, where they go to school, but we can actually use data linked to addresses of all those places that are important to our lives And we can actually create estimates, very reliable estimates, by taking advantage of, in the lower left-hand corner of this slide, it's a map of the Northeast United States, and all the dots represent ground monitors that are set up and monitor daily pollution levels, different components that are set up by the U.S. Environmental Protection Agency. And you can see that, you know, they're pretty dense in certain areas where there's a lot of population, and they might not be so dense otherwise. And we need to sort of think about that and accommodate that in the modeling approaches that we take. And then they use NASA satellite data in the upper left-hand corner that gives us other information that helps us contribute that to the information that we're feeding in to develop these models. So we have a whole host of folks who have particular expertise into developing models. One of the first models we were able to do, as I said, was PM 2.5, because it's regularly monitored with those ground monitors from the EPA. Now, how our science helps change policy and change what those monitors are measuring is we start in other ways to measure things that show, hey, this might be driving health even more so than what you're already measuring. So we need to invest and think about doing that. And the science over time has contributed to growing what those ground monitors are actually measuring at the EPA. So that's kind of a policy level way that we might have effect. So as I said, we actually map those estimates that the scientists create for different air pollution components, and we map it to where you live, where you go to school, where you work, depending on the questions we have. Now, the nice thing about that too is, as Wendy mentioned, the other thing about exposomics is we want to understand not just what you're exposed to today, but we want to be able to recreate as much as we can back in time to really measure as much of your lifespan as we can when we're actually looking at these data to see how they might in fact affect health. Well, satellites went up at different times, 2000, 2002, 2003. So as long as we know addresses over time, we can actually recreate your exposure to these things all the way back to up to 20 years or more at this point. And we do it not only for air pollution, and we'll come back to the fact that we can link this to lots of different outcomes, including sociodemographic factors. Now, what do we do with that data? Now we have data, we can estimate daily exposures. Daily exposures, can you imagine, at where this person lives. So take a residential address, for example. And on the bottom left-hand side of this slide, this actually, these squiggly colored lines map actually real life exposure in some of our select women that we've recruited during pregnancy in the New York City area. And this maps basically your air pollution exposure levels to the PM 2.5 over the entire gestation. So over 40 weeks of pregnancy is what that is looking at. And I have that below a map of what we understand to be the developmental processes involved in that growing lung that starts in utero. If you go to the left-hand side of that cartoon picture up above in the upper left-hand corner, it shows how the lungs develop, those airways develop over time, and the different other related processes that go along with developing the lung so that you can start breathing the air once you're born. And you can see there's very rapid development over pregnancy, and then by the time you're born, there's still, the interesting thing about the respiratory system, it continues to grow and develop until we're approximately end of adolescence, early adulthood. It's a little bit different for different people, a little bit different based on gender, that kind of thing. But so it's an interesting system to think about from an exposomic point of view, because that and those environmental influences are going to have a long time to have effect on that system. But the most important period is that prenatal period in the early first 2 to 3 years of life. Because by the time we're 6 or 7 years old, our level of lung function at that age tracks for the rest of our lives. We're going to be, if we start lower than others, we're going to lose it faster over time, and we're going to be more susceptible to developing chronic lung disease. So if we can think about and understand what all the environmental influences that optimize a person's lung development in that early period, and how do we protect against things like air pollution, that is going to be very, very effective to preventing long-term, lifetime problems with the lungs. So here's a reason why I want to, if this next few slides, show you some results. It's going to be pictures using that daily air pollution exposure over pregnancy now. So we're talking about what moms are exposed to over pregnancy, and then we're going to look at outcomes for respiratory disease development in children. And here we're going to look at asthma diagnosis by the time kids are 6 to 7 years old, predicted by what mom was exposed to in pregnancy. And here again, we're just looking at one pollutant, it's PM 2.5. So we're looking at this squiggly line base over time. When it goes above the dotted line, that's where there's a significant relationship between increased exposure to PM 2.5 in this window that has now circled in the blue, predictive of increased risk of asthma in kids. So if you were in utero exposed to this, you're more likely to develop asthma by the time you're 6 or 7 years of age. That happens to be in the middle of the second trimester, and we can link that back to that map I showed, the cartoon of the developing lung, to understand what mechanism might be operating there. Well, that's interesting. That's something we found about 6 or 7 years ago. And then we actually said, well, why don't we look at a different outcome related to the same pollutant over that gestational period? And you can see on the left-hand side, with again circled in blue, it's not the middle of pregnancy that's increasing risk for low levels of lung function by the time those kids are 6 to 7. It's right at that late pregnancy where the hit is happening, where the kid has lower levels of lung function. It dips below that line. And that says those are the kids that are at higher risk of developing reduced lung function. Now, again, as I said, your level of lung function at age 6 to 7, which we're measuring here as the outcome, predicts what your lifelong lung function is going to be. So already those kids are behind the 8 ball. But it also is interesting because we know there are going to be different critical windows where that exposure is going to matter for various outcomes. That's another reason why we want to have that high temporally resolved daily measurements of these air pollutants. So these tools that we've developed to do that and give that kind of data are going to be very informative. Now let's take another pollutant. Let's move away from PM 2.5. We've also found nitrate. And that, when we took a look at that in our model, in predicting reduced lung function is where it's circled in the blue. And again, boys and girls, we're not going to really talk about the sex-specific effects, but the reason I'm showing boys and girls is a little bit different for the sexes. It's a different window. Remember, PM 2.5 was at the end of pregnancy. Here we're seeing an early window of time where they're more vulnerable to this particular air pollutant. So it's operating through a little bit different mechanisms. So it's affecting different pathways and processes that are related to lung growth and development for that pollutant. So that says we need to measure more than one pollutant at a time and be able to look at those in our models. But then we're also seeing data from animal studies that said, well, the smaller, if you look at the left-hand side of this graphic, and I'm looking at the boxes where they have the different sizes of air pollution particles. Force level less than or equal to 10 micrometers. The fine PM 2.5 is less than or equal to 2.5. That's what we've been talking about. But now you can measure even smaller particles. The science advances. We can measure smaller particles, ultrafine particles less than or equal to 0.1 micron in size. And you can see that how does air pollution get in the body to affect our health, whether you're talking about a pregnant woman that starts to affect the fetus or a non-pregnant person where it's their own issue with what they're breathing. It gets into the lungs, it translocates through the lungs, through the alveoli, into the bloodstream, travels around our body and can affect different processes and have impact on problems not only locally in the lung, but more systemically as well. We know the smaller the particles, the more toxic they can be, and we need to be able to measure them. Well, EPA doesn't monitor ultrafine particles, So we went about measuring and linking it to some of our outcomes to sort of start to say, hey, this is even more toxic than the others. We need to push you to start to monitor this and regulate this just as you do the other size particles. And we had a paper that came out just a year and a half or so ago that showed, again, ultrafine particles were associated with increased risk of asthma in the kids by the time they were 6 or 7 years old. And it was late in pregnancy where that seemed to be most impactful here. But now we know, okay, we can measure all these different components of the air pollution. We know that different ones are going to be differently toxic if you look at them one at a time, but we might be missing information if we don't actually think of them as a mixture where we can actually measure, not only measure, but consider them in our analyses together as a mixture, because the other pollutants that you travel with are going to change the potential toxicity of a given other component, right? We're not exposed to one thing at a time, but we're exposed to kind of a soup of all the different multiple components at once. And even at low levels, when you have multiple pollutants pushing and pulling on some of the same, change the biology in similar but maybe not completely overlapping ways, that where you start to see multiplicative effects of the mixture of these things. So we really pushed our analytic scientists to say, well, okay, we're gonna go after measuring all of these different components of the pollutants. We want you to help us develop methods to where scientifically we can actually examine those collectively together. And we did that for the Northeast, which is what the map shows. And we looked at multiple constituents of air pollution. Now we don't look at one thing at a time, just nitrate, just PM 2.5, but we can actually look at all of these different pollutants on the left-hand side and the word cloud and actually put them through our model over gestation, still have daily measures of all of these things, just as we've shown for some of the single pollutant models. And now we can say, well, what's important? What's the most important driver in Boston in this community versus New York City in that community or other places around the United States, et cetera? It's going to be different for different pollutants because the composition varies based on the weather, the humidity, all these climate change or climate related factors that we know are going to be important to think about with climate change. And we need to monitor it over time because it is, we've seen already what's happened to climate, and it's only going to continue to change more and change that distribution and change who we think are going to be most vulnerable in those particular regions. And this is the first study, and I'm showing you data now that we have out under review, but it hasn't been published yet, where we actually looked at a mixture of 7 different pollution components that we've measured now that are collectively available through the Institute to start to link to some of these outcomes. And we linked it to lung function at age 7 years again, similarly as we did with the single pollutant model. And actually, when we put them all in the model, it wasn't PM 2.5, it wasn't nitrate that came out, but it was ozone and that we're seeing here. And then we looked at it by different race ethnicities to see if they were differentially impactful. The mixture of pollutant was differentially impactful on certain populations. And we saw a greater effect in African American and Hispanic in particular, and Hispanic in particular for this particular population. And Wendy also alluded to the fact that we also measure not just pollution, but we measure social demographic factors and social stressors and other things like nutrients and diet, nutrition, access to healthy foods, these kinds of things. We're digging a little deeper to say what is really driving this differential effect, but a lot of it looks like it's going to be poverty, exposure to greater stressors, et cetera, that enhance the impact of these air pollutants. And we've shown that. We won't have time to look at a lot of the information, but we have to start thinking about not just multiple air pollutants, but then how do we do a mixture with all these different other social factors, et cetera, in them as well. Now, what we've talked about mostly is a more public health kind of approach. How do we find out what constitution of different environmental exposures at a vulnerable developmental period of time, like pregnancy, relates to disease onset like asthma in early childhood? And why are we doing that? Because we want to prevent. We want to be able to say, you know, okay, maybe you're living in an environment with these multiple pollutants, but maybe if we can enhance your access to healthy foods, et cetera, that we know can actually mitigate and dial back the impacts of the toxic pollutants or metals or chemicals or whatever we're looking at together as a mixture, maybe we can actually, lead to prevention of disease. And we will, and I think we've had some insights into that already. But how about kids who already have disease, or we're talking now about people later in life, adulthood, now they've had chronic lung disease, and now they're coming in to see me. Could we think about a scenario where understanding this context is going to be very important to how we manage their disease going forward, and think more about how exposure to these factors impact treatment of disease, your response to therapy, your potential perhaps to have side effects of that therapy, et cetera. And so we're trying to move forward to think about and assess disease progression in relationship to exposomics. And here, particularly with climate change, we're worried that there's going to be increasing exposure to air pollution over time. We want to monitor that, and we want to look at these mixtures and how not only will it lead to onset of the disease, but how about accelerating disease severity in people? We see this happening in medicine. People have been scratching their heads about this. It's really likely to be some of these environmental factors that are changing over time that do this, and again, can affect treatment response, as well as early, perhaps leading to earlier mortality related to the disease if we can't pick up a signal earlier on that says, hey, this person lives in an environment where they're increased exposure to lots of these different toxins. We need to do something to get them back on a more optimal path. So we can do that throughout the disease, natural history of the disease prior to onset. That also has implications once you do have the disease when you're thinking about it clinically. And I'm just bringing this back to say we've talked about air pollution in particular and just how complicated thinking and measuring and characterizing one's exposure to that. We're so far down the road in a relatively short period of time to be able to measure multiple air pollutants together, have developed the computational tools to be able to analyze those together and get more precise information about what might be driving that. At a community level now, will we ever get to that personalized individual? Probably not, but we're getting closer to being able to put people in context to really understand that we might need to think about and manage their respiratory disease differently based on that context. But I also put this here to remind ourselves that I've only talked about the one, but really our models are also considering mixtures of all of these different factors, which we know are going to be only increased and enhanced through climate change going forward, with particular communities at risk for different sort of proportions of what might be going on there. And so if we can start to measure it, build platforms where we have all this data on people, we'll be able to be more responsive to keeping them healthy. So I always think of the elephant, the fable of the elephant. You have people blindfolded, touching different parts of the elephant. It's like if you only think of one chemical at a time, one factor at a time, we get a different answer. We might get the wrong answer. We might miss totally what's going on. Everybody touching one part of it interprets what they see or feel or touch. And here's what we have to do, and this is what exposomics is. It's really putting all of the pieces together and thinking about how do we not only measure, keep track of this for people that we're taking care of in our health system, but also have the tools to be able to analyze it, ask and answer questions about it, so we do kind of understand the whole person as they sit in front of us holistically, the whole person, the whole elephant. And what are we doing to be able to do that? We're building that platform. I've talked to you about a very small portion of what we're putting on that platform here when I talk about the air pollution modeling that we're doing, coupling that with the EPA air quality monitors, for example. But we're also doing it through measuring wearables, having people wear wearable sensors and devices that can measure exposure to hundreds of chemicals at a time, your biological response to that through some of these wearables, et cetera. Also using things like smartphones, smartwatches, crowdsourcing, et cetera. But it's all to get multiple layers of this geospatial data linked to where you live, work, go to school, et cetera, that we can then pull together with things like our healthcare data warehouse, our electronic health record, and look at how it's impacting our populations, being able to map, say just for Mount Sinai's health system, being able to map across the boroughs of New York City and show the hot spots and what's driving it, that kind of thing. And we're already starting to do this and seeing, it's just really fun to see what's been developing over the last 5 years even. But it takes a lot to build and integrate all of these different components of the platform. And that is something that we're very much working towards. And actually, Wendy mentioned I'm the Dean for Translation of Biomedical Sciences for the health system. That means I also lead what's called our Clinical Translation Science Award. It's infrastructure funding from NIH. We get about $7 million a year. And a lot of that is being put towards developing this ecosystem that will allow us to bring some of the work we're doing at the Exposomic Institute more into the translation down to communities and patient levels. So we measure lots of different things to do that, and this just exemplifies we do more than just air pollution. We're also funded as part of this national consortium, ECHO, Environmental Influences on Child Health Outcomes, across the country. So it links not only the cohorts we're following, the populations we're following in the northeastern United States, but you can see we're linked and collaborating with lots of folks who are also following similar populations across the whole US. And there we can do even more impactful science because we've extended our models, our air pollution models, across the entire United States. And so we can just really look across different regions and see what the different drivers are of the different outcomes. And respiratory is a big interest across that consortium. And this is the idea. It's really, one day as a clinician, I'm going to be able to fine tune people by actually having this kind of information at hand on a very wide scope of what are the important contextual environmental drivers of maybe this person sitting in front of me, and how does it change how I might think about their disease, treat their disease, prevent their disease, et cetera, from becoming more severe and leading to things like early mortality. So hopefully that's given you some high-level concepts of how, as clinicians, we think this is going to help us in the long run. And also how far we've come. I mean, some of the work I've shown you here is just phenomenal, how fast we've been able to develop some, expand the number of models when we're talking about just one thing, air pollution components and constituents, et cetera, and being able to translate that to some of these respiratory outcomes. So I thank you for your time. I'd have gone a little bit over, but we'll have time for questions. And I'm also happy to answer questions ongoing in other ways.
Well, thank you so much, Dr. Wright, for all the important work you're doing. And just to let everyone know, we're going to go a little bit later, maybe 5 minutes for extra questions and answers. But for now, I invite everyone to visit our website, MountSinaiExposomics.org, for more information and resources about how the environment impacts our health. Also, be sure to follow our social media accounts at Sinai Exposomics to get real-time updates. And if we're unable to answer all your questions today, we'll work on getting those answers to you on our social media platforms. And if you think of any additional questions after the conclusion of this talk, please, or have specific environmental concerns in your own community, from questions about pesticide use in public parks to the safety of your water supply, our scientists are available to speak to you and provide the data that underpin sound policies that promote healthy environments. Please don't hesitate to contact us. And as a member of the Exposomics and Children's Environmental Health Board at Mount Sinai, I invite you to get more involved. We welcome nominations for the board, which drives the philanthropy underpinning the work that Mount Sinai does at the Institute for Exposomic Research. Please contact us to nominate someone and learn more. Okay, so now for the Q&A. For the first question, I know Mount Sinai trains pediatricians to understand environmental exposures for their patients, but have you explored training OBGYNs?
We have talked about, just as I showed you, and I'm a good example of it. I'm an adult trained pulmonary critical care physician, and I sit in the pediatrics department. And one of the reasons I came to Mount Sinai and I was excited to join a pediatrics department is they were already doing a lot of it for training and thinking about it in pediatrics, but we need to back it up even further. Look at OBGYN, look at adolescent medicine physicians, et cetera, because we know your health is important as you go through your life course. And if, particularly for women who are going to carry infants and we know the impacts on the fetus, not to put all this on women, but we know it's a critical window. So the more we can make you healthy as an adolescent going into adulthood to where you're going to be, going through these different life phases, we can have so much impact. So absolutely, yes, we're talking about not only OBGYN, neonatologists, adolescent medicine physicians, adult trained physicians, whether you're gastroenterologist or what have you. We're talking to them all and educating them around the idea that they have to think about where people have come from to understand how to best care for them now, I guess I would say. That's a very important question. That's a very important part of what we do as well and think about in pediatrics. One thing we have, and we were blessed to be able to get funded through National Institutes of Environmental Health Sciences, is actually a training program specifically for OBGYN, folks taking care of people in the prenatal period, and neonatologists and pediatricians to start this early life training as clinician scientists, or those who are translating it to health.
And related to that,
we need more of that. Yeah, sorry, go ahead.
Oh, and just related to that, are there specifically about foods and nutrition, are there specific foods that pregnant women can eat that improve outcomes for their children? Or what about all of us? You mentioned fish and plant-based foods, and I'm just wondering if there's specific foods. What are your top few? Yeah,
I mean, the data now, and some of this is our data coupled with, lots of folks with antioxidants mitigate effects of not air pollution, a major pathway through which a lot of these air pollutants lead.
We're losing your volume a little.
Can you hear me? Okay.
Okay. You're in and out a little.
Okay. Let me hold. I'm sorry, I have to do the phone. My audio on the computer somehow clonked, but is that better?
I hear you now.
Okay, I'll hold the phone up. So the data now says antioxidants, right? We know that if you know the mechanism of a lot of these air pollutions, it's through enhanced oxidative stress in the body. And that can have adverse effects on the lungs, but other areas as well, brain, etc. So increased intake of antioxidants. A lot of that's in green leafy vegetables, those kinds of things. The less red meat, the more you lean towards the green leafy vegetables. We're supposed to have so many fruits and vegetables a day. Antioxidants. Other fruits, for example, blueberries are very high in antioxidants. So just have knowledge around that and think about what we're eating. But I mentioned the polyunsaturated fatty acids. There are good ones and bad ones. There are pro-inflammatory and anti-inflammatory, and it's a balance. Fish, there are certain seafoods that give you higher levels of the anti-inflammatory polyunsaturated fat acids. We need all of it, but you think about what, how you want to tend towards having more of the better ones that are seemingly helpful. So, and I think nutrition is something that has to make its way back into medicine more than it has in the past, right? It seems like it's separate. Someone else should be talking to you about your diet. Well, we as clinicians need to be more knowledgeable about that too, so we can provide that information to our patients, or at least have ancillary teams that can do that. I think that's a great lever. Another one is, besides diet, is stress reduction. How can we think about that? And that can be very tailored to individuals, to communities, to et cetera. But we need to be talking about that as clinicians with our patients and thinking together about that as well, because that can mitigate the effects of a lot of these chemical toxins as well.
Thank you for that. Are you evaluating indoor air exposures? They are not directly related to the outdoor environment.
Yeah, that's an important part of air pollution. Here, what I've been talking to you about is outdoor air pollution exposure, and it's not the same, right? It depends on how open your home is to how equilibrated that is with the outdoor environment. But we do measure indoor environment. We measure it specifically for studies we're doing, for example, around sleep, because you're sleeping in your home, and we want to know not only exposure to things like light at night that might disrupt your sleep, noise at night that might disrupt your sleep, but also the pollution can have those impacts as well. So there we do it a little differently. We have a monitoring device that goes right into the home, and we're measuring over a period of time that makes sense, et cetera. We need to do more of that. There are measures, there's equipment that's being developed to make that more readily accessible, right? If you have a small wearable device that someone can wear and walk around outdoors during the day, go to school with, come home, sleep with, those things are going to give us much more data over the whole day, and we'll get even closer to what's driving health in these folks.
Amazing.
That's a good question.
Thank you for that. And you touched upon communities of color. And just could you tell us more about how air quality is related to the health inequities that we're seeing?
Yeah. So lots of things that might drive air pollution, like the types of traffic that you have going through your community, maybe industries in the area that are polluting the air. Those are placed, they're not randomly placed in different areas. So there are certainly issues related to sort of historical decisions that were made around those kinds of things that make some historically marginalized communities be more burdened by poor air quality because of this. And we know this is happening upstream. We have to take into account and try to measure that as much as possible, and think about that as it might be explaining some of these health disparities. I did show you the impact of the air pollution mixture was different in communities of color, right? We saw more in the Hispanics and the African Americans compared to the white non-Hispanic cohorts. A lot of it, some of that's due to differential exposure to the air pollutants themselves, but it's also, as I mentioned just briefly, other things that track with it. They might have more stress, right? More poverty, and lots of stressors come with that. They might have less access to healthy foods, so their diet and nutrition, they have more inadequacies in some of these key micronutrients like like the antioxidants, like the PUFAs. And we're trying to look at it all together, because it might be that this community could really benefit and have less impact of things like air pollution if we can really increase their access to healthy foods, et cetera, and education.
Right. Well,
around
thank
that.
you. Thank you so much. We ran a little bit over, but thank you, Dr. Wright, and thank you for everyone who joined us today. We have an evaluation survey. If you could please fill that out, it's in the chat, and we'll email it to everyone who registered. And next Wednesday, we'll have a conversation about infectious disease in the environment with Dr. Nick DeFelice. And thank you so much, everyone, and have a great day.
Thanks, everyone.